lunch. Because the virtual nature of this conference this year, because of COVID, we will have lightning talks instead of our usual poster session. These lightning talks are going to be each 4 to 5 minutes long, and we have pre-stitched them together into a couple longer videos for each of the 2 sessions, one for each session. There are 6 talks in this current section and 5 talks in the second session later on this afternoon. I'll be moderating the question session at the end of each of these, and I encourage you to type your questions in the chat as they occur to you instead of waiting until the end, just so we don't have too much of a lag between the question period and the talks. If there are too many questions allotted for the time, you can continue to answer the questions in the chat as the meeting continues. And it seems that there are some conflicts between maybe Office 365 and Zoom and the particular way that this meeting was set up. And so we will not be able to unmute you for any of the questions that are being asked if you have questions that you need to ask out loud. So we will have to rely on the chat box to do all the question and answers. The speakers will answer out loud, of course. Let's see, so we also have some judges that will ask questions, and these judges will rank the speakers based on the speaker's delivery, content and organization, their take-home message, responses to the question and answer period, and their ideas and how innovative they are. The 3 best lightning talks across both sessions in total will be awarded with gift cards. And with that, let's begin the first session of our lightning talk. Why don't we, in the meantime, try to go to the next speaker, and maybe someone can help Amelia somehow unmute herself. This might be a technical difficulty. So for now, let us please try to talk to Ed Grow. Ed, are you able to unmute yourself?
Everyone should be able to unmute themselves now. Let's try that.
Okay, let's try round 2. Technical difficulties will be gone now, I'm sure. Let's try Amelia again. Are you able to unmute yourself?
Yes, I am unmuted now.
Thank God, I didn't want to answer your questions for you. Okay, so the first question from Suma. What is the criteria you used for selecting the genotypes in this study?
Yeah, so just being able to answer both questions in the chat. They're very similar answers, so I will just answer both of those together. The FR697 seedling root system is able to maintain growth in dry soil. So that was how we ended up choosing FR697, and this was superior to B73, which is how we went into the choosing of these genotypes.
Okay, great. And you answered the question about the seminal and primary roots of B73 versus FR697.
Right, and those mechanisms aren't exactly the same in the primary roots versus the nodal roots, because the whole seedling system would be drought stressed. So there are additional mechanisms that would be involved in that process, but we're looking just specifically at the nodal root mechanisms.
Okay, great. So the second question is one that had been asked by Norman about comparison to the nodal roots. So a second question from Suman that I received earlier. What you expect, I'm assuming this means, do you expect anatomical significance to avoid nodal root dehydration in the FR697 genotype?
Yeah, so that's something I've been working on looking into right now. I'm looking at suberization in the outer portions of the root. We are thinking that there may be a difference in where suberization begins along the root, and this might start earlier or later between the different genotypes that allows it to maintain its water status and water potential.
Okay, great. And here's a general question that probably most of the speakers will be asked. So this is one that's kind of more retrospective. Many times as you reach the end of your project, you find things that you wish you would have changed along the way. So if you could restart your project, what would you have done differently? That question comes from Kristen Billieu. There's your video, great.
Yes, so something I would do differently. I would like to look at these maize genotypes grown out in the field and be able to compare directly to the roots grown in the lab. And that would be able to directly see the difference and make sure that the results that we're finding in the lab are also working in the field. And this would involve a little bit more and is being done by some other members in the lab. But I would want to look specifically at the two genotypes that I'm looking at.
Great. All right, we'll see how the timing works here, but I think for now we'll move to our next lightning speaker. So Jed Groh, would you please turn on your video and or your microphone, whichever works best for you?
Yes, can you guys hear me?
I can hear you wonderfully. Thank you. Again, thank goodness this is working out. I'm starting to panic just a little bit. Okay, so we have a number of questions for you. I see you must be in a common space. You have your mask on.
Yes, yes. Yep.
So from Suman, how are these advanced lines better than the checks with respect to chipping qualities?
So I mentioned they don't have to undergo storage to help the sugars be converted back into starch. They chip well, so that's a benefit as far as chipping quality goes. Overall, their specific gravities, which is the percentage of dry matter content compared to the percentage of water. So how much is it squishy and how much is it that hard stuff that chips really well. Overall, they were a little lower. We think it had something to do with precipitation that year. I mentioned in my paper that the precipitation overall for these 2 years seemed to be a little higher than compared, which was nice for yield. But as far as specific gravity goes, it's a little lower. We give them a Hunter L score, which is kind of how light or dark they are. And they chipped fairly well. It was actually a little better color than the Dakota Pearl. And that might have to do with the fact that they were chipped directly out of the field, or could be because of that lower sugar levels overall. So they chipped fairly decently. A lot of people seem to like the color, processors, so more than what they currently have, but that's subjective.
Okay, great. Another question from Suman that I received earlier. The graph shows that there's not a difference in sucrose content between the Czech and the new North Dakota 7799C1 during the storage after 6 months, which contradicts your talk. Could you clarify, please?
Yes, so as far as statistical differences go, you could say there was one with an alpha level of 0.05. The problem is, and why I didn't include this, is because they have thresholds in the processing industry. So it doesn't really matter as much compared to, is there a statistical difference between this and this? They just matter what difference, what level it is. So if it's greater than 1 milligram per gram, they won't even take it. And so it's because of these threshold dates and these graphs were submitted to the industry that they're really not important as far as statistical differences between cultivars. So yes, there was a difference. We didn't actually analyze it statistically because we were looking at that cutoff level. So good question.
Okay,
I'm glad you caught that. I'm glad someone's looking at them that closely. So
Norman asks, what do you think would be the most effective way to influence farmers to change their management practices and reduce nitrogen inputs? And I think a related question to nitrogen from Eduardo is, what are the organic matter levels in the field experiments? And maybe this has to do with nitrogen as well.
Yeah, so the organic matters, as far as North Dakota go, are pretty high. We have Williams soils, is what they call it. They're nice organic levels. Particularly in these fields, they were about 5.9%. So nothing spectacular. It could be anywhere from 2 to 10% usually when you're looking at organic metal content. These had winter wheat grown on previously, so nothing out of the ordinary for the area. As far as getting them to decrease the nitrogen levels, that's a key. And we keep breeding these cultivars with lower and lower nitrogen requirements so they can use less nitrogen, but still get this increase in yield. And hopefully hey, you guys can use less nitrogen and then there's this positive benefit. But it's got a lot to do, there was another question about extension, and it's got a lot to do with that. Because we can show them that, well, you don't need an increase in yield, but it's really that trust factor. I can say, hey, why don't you decrease your nitrogen by 100 grams sorry, 100 pounds per acre, and you'll get the same amount of yield. That's a huge risk involved for the farmer, because especially on dryland conditions, I mean, you're totally dependent on moisture and you're totally dependent on your nitrogen. And so if you don't, if you do it, decrease it, and you see a lot drop in yield, it's not my paycheck that's going to take a hit, it's yours. And so there's a lot of trust when it comes to that. And so a lot of these farmers actually paid into this experiment, paid the school significant amount that they says, okay, why don't you test nitrogen levels for us with these new cultivars, and we'll really see if we can decrease it. So we're hoping that this study gets through the extension. We're able to really let them know that there's this benefit. So
Great, thank you so much. We will move on to the next speaker, Letícia Inoue. I hope I'm pronouncing it.
Inoue.
Inoue, okay, thank you
Yeah.
for correcting me. I appreciate that. We do have several questions for you. One of them is from Norman. He asks, why did you choose vermiculite and sand as your substrate? Do you think the results would be different in a field setting?
Well, we use vermiculite to facilitate root removal completely by washing with water and with minimal losses. The substrate was corrected with nutrients so that they were at critical levels required for the plants. If you use another substrate like soil, many root losses could be reported as they break easily.
Okay, great. A question from me. Do you think your results will be applicable to soybeans across the world, or do you think that there's something unique to the varieties that you tested or your environmental conditions?
The variety that I tested was Brazilian variety, so we needed to make those tests with another varieties around the world. So that maybe this results is for Brazil, because our environment conditions that is different of the other countries around the world.
Okay, great. And then one more question. This is the more philosophical question for you. If you could go back and restart your project, what would you have done differently, if anything?
Whoa, this is a difficult one. I need to think. It's a difficult one.
Well, perhaps you're very happy with your experiment and there's nothing to improve.
I think it's a better place to work, maybe because I had some difficulties with some people when I work, but it's my thing. It's not about the research. In the research, there was nothing that I would change, because I had the help of my advisor and my co-advisors, and they helped me a lot. So I am very glad for them. And yes, I think it's not in my research that I could, if I could change anything, I wouldn't change anything.
Oh, well, that is a great answer then. Thank you.
Thank you.
All right, we'll move on to our next speaker, Ankith M. Ankith M.
Yes, I'm here.
All right, great. We can hear you and we can see you. Wonderful. Thank you. So here's a question from Suman. How would you confirm that the E. coli cells had the gene of interest before doing the expression analysis of seed coat protein? For example, the gene of interest.
We have done PCR for gene-specific primers, and also we have done restriction digestion by expand that set.
Okay. And then I think a related question from the same person, Suman. Have you checked the copy number of the gene of interest in your E. coli cells? Does the presence of 3 bands in your SDS gel, are they transcripts of one gene?
3 bands. No, actually it is only one band, distinct band. So in the 34 kilodalton. So I didn't check for the copy number of the E. coli.
Okay. Then there's a question from Jianbin. Would you like to try mRNA to produce your antibody like the COVID vaccine?
Can you repeat once, please?
Would you like to try mRNA to produce your antibody like the COVID vaccine?
Yeah, I would try that thing because of the it will have a regulation, and it will have certain high level BSL-4 requirements. So I could not do.
Okay, and then I will ask you the same question about your project. If you could restart your project from the beginning, what would you have done differently, if anything?
Maybe I will develop some CRISPR for the detection of the thing. So now I used ELISA for the detection of the disease. So I will use CRISPR for the detection. So.
Okay. Great.
Okay. Thank you.
Thank you very much for being here. Our next speaker is Bruna Montes Luiz. And we have several questions for her. Some are typed in the chat and some I received earlier. How and at what soybean growth stage that the plants were infected with transgenic Bradyrhizobium?
So in the case of the last experiment that I counted the nodules for, we actually generate transgenic roots. So it's kind of a weird process because it's not exactly the same as a normal growth. We actually remove the roots. So they're about 20 days old, around 20 days old when they are inoculated, but they have been through some torture, let's say, because we cut off the roots, we add agrobacterium to generate transgenic roots. So it's not a normal growth for 20-day-old seedlings, and then they're inoculated and the nodulation is assessed 4 weeks later.
Okay, great. Have you tried applying GA to the RNAi GA3BDO to see if it recovers the phenotype and increases the number of nodules?
Not yet. So what's happening at the moment is I'm still working on these experiments. I've shown 3 results for you, but I have about 40 constructs that I'm working on, and I have 3 replicates for each. So it's a time sensitive. So it takes a long time for each experiment. And so I haven't gotten to the point of analyzing the effects, but that would be the first step in assessing the phenotype for the GA line.
Okay. Here's another question. Do you have a stable transgenic soybean line? Do you have a stable transgenic soybean line expressing cortex promoter driven RPL18- flag? And did you do flag IP RNA-seq yourself?
So first question is if we have a stable line. We do not. We did this via hairy root transformation. So it's only the transgenic roots and not the whole transgenic plant. It wasn't me. I wasn't the one who did this. This has been an ongoing project in the laboratory involving 3 other postdocs. So I wasn't the one who did that part, but we're working now on a phloem specific experiment. This one was cortex.
Okay. And then here comes the philosophical question for you. Is there anything that you would want to redo differently if you could restart your project?
I'm thinking maybe in the timeline of experiments to focus on having our 3 replicates at first, one after the other, because I first did a major screening of all of them, and the ones that gave me results I've repeated and I'm repeating. That's the only thing I can think about. Also keep in mind that this is an ongoing project. I'm still at the beginning of my PhD now. I've been working on this for a year and a half, so there's still some work left to be done.
Okay, thank you very much for your time.
Thank you.
And now we'll ask questions to our final speaker, Darshana Parta. If you can turn on your microphone and your video if you wish. Hello.
Hello.
Good to see you. Okay, so here are some questions. Did you identify any specific genetic factors that were similar between the elite genotypes that clustered together?
Yeah, I had clustered some of the groups where I found that the grouping was based on some of the similar characters based on the nature of the morphogenetic characters. Like in the first group which I had clustered, it was short fiber crops and it had high seed oil content and the seed weight was also higher. And the last group was a dual purpose crop and it is one of the Czech variety. And I think maybe because of these common genetic factors, they were getting together as a cluster.
Okay, great. And then I'm going to show my ignorance about flax. Can you tell me about the center of origin of flax? I don't know if flax is an open pollinated species or a selfing species. How diverse is the germplasm collection? And are the varieties that you tested, were they populations or inbreds or hybrids?
Actually, first of all, the center of origin is also, it is a confusing between two group of thoughts. Like some say it is from Iran, some say it's from Egypt. And coming to another question that you were saying, the genotypes which I had taken, those are open pollinated varieties, and the center of origin, regardless of that, we are having some of the center of diversities also connected to the, where we are cultivating it. And it also depends on various factors, like on which purpose you are doing the crop. Like flax and linseed are both interchangeably the same term. So some say as fiber flax, and if you are using for seed oil purpose, it is called as linseed. But because my thesis work was on a dual purpose crop, so I integrated both these concepts, and the biochemical analysis which I did, it was basically upon the oil quality, which is good for our health, because due to oxidation of the oil, auto-oxidation of the oil, it is affecting the taste and aroma of the oil. So that's why the marketability is also decreasing.
Okay, wonderful. So it's almost like they're two different crop species altogether. Okay. And then here's the philosophical question for you. If you wanted to restart your project, what would you have done differently?
Yeah, exactly what I was telling now. First of all, I would have sorted out this because flax or linseed is having a narrow genetic base. So within 2-3 genotypes which are from one state, or the genotypes, they are having not much variability. So I would have sorted out the distinct genotypes which were having linolenic acid less than 5%. And based on that, it would have industrial benefits also. So by sorting out out of 52 genotypes, I would have taken less number of genotypes and started my work, which would have been more profitable and less confusing for me.
Okay, less confusing is always better. All right. Thank you very much for our first 6 speakers in the lightning session. You guys did a great job uploading and answering your questions and everything. Your research projects are all very interesting. And so with that, I will close out the lightning talks part of this program. The first part, we'll have another session that begins around 3 o'clock this afternoon for the next 5 speakers. And so with that, I will pass it off to our next section, where we, let's see, Zhonghua will introduce our next speaker. Thank you.